So lipid nanoparticles, or LNPs, gain wide public attention as part of the mRNA vaccines for COVID-19. However, LNPs have been used and are continuing to be used in a wide variety of pharmaceutical applications, such as for other viral vaccines, cancer therapies, analgesics, and other therapies. And some of those are shown in the diagram here. So the size of lipid nanoparticles affect the delivery efficacy. Smaller LNPs more easily penetrate tissue and deliver mRNA. And one of the reasons this is important is because during vaccine production, there's a low yield, and that means that very high doses have to be given. So anything that improves the uptake is important. Now, LNP size can be controlled via mixing time. In general, longer mixing times give lipids more time to aggregate and hence lead to larger aggregates and more heterogeneous size distributions, while shorter mixing times lead to smaller nanoparticles, but at the same time, lower yield. And we'll talk more about mixing later in the talk. So precise tuning of the LNP size by experimental iteration is often done, but it's very time consuming and therefore costly. And this motivates the need for simulations to complement experimental development. Multi-physics simulations of the flow, transport, and LNP self-assembly can improve the understanding of the LNP formation. They relate key outcomes such as the LNP size distribution with system parameters for design, fluids, operation, lipids, etc. The goal is to reduce the number of the required costly experiments and increase the value of each of those using simulation. And simulation guides the design iteration. It can suggest new avenues for development that may not be obvious from empirical intuition alone. So in today's talk, we'll outline the modeling, physics, and workflow required to model mRNA vaccine production. And in particular, we'll review turbulent and laminar mixing, fluid flow and solvent transport, LNP formation and self-assembly, and population balance modeling. We'll then discuss case studies on chaotic mixing and microfluidics and the coupled mixing and LNP self-assembly.
So now let's get into some of the key physics and modeling strategies using COMSOL. So mixing can occur in essentially two flavors in production. There's turbulent mixing and there's laminar mixing. In turbulent mixing, mixing occurs via the breakdown of very large eddies into smaller and smaller scales. Can be further subdivided into three regimes, so-called macro, meso, and micro scale mixing. Typically, macro mixing is not rate limiting. Meso or micro mixing can become rate limiting. And in micro mixing in particular, there are two timescales of relevance, depending on the so-called Schmidt number, which is the ratio of the kinematic viscosity of the fluid to the scalar diffusivity of the thing being transported and mixed. For moderate Schmidt numbers, you are in a Kolmogorov regime, and the mixing is dictated by the Kolmogorov timescale of turbulence. Larger Schmidt numbers, the mixing is dictated by the scalar diffusivity, and that's given by the Batchelor time. Laminar mixing is a completely different beast. So laminar or microfluidic mixing occurs in microfluidic devices, and there you don't have the breakdown of large eddies, and instead you are relying on molecular diffusion for components to mix between streamlines. The disadvantage here is that molecular diffusion is very slow, and so commercial microfluidics typically employ chaotic mixing strategies in order to overcome the slowness of diffusion. We'll talk about that in some of the case studies we're going to discuss. The way these processes are modeled in COMSOL is using a combination of the fluid flow modules and the transport modules. So the fluid flow modules essentially involve the solution of the Navier-Stokes equations for the velocities and the pressures in the fluid. But since we have typically two solvents, these fluid flow equations, which apply to both the turbulent and laminar cases, must be coupled to transport equations for the solvents. And since they are in a concentrated regime, the Maxwell-Stefan equations are the appropriate transport equations for the solvents. So the important thing to keep in mind here is that all the properties of these fluids, the densities and the viscosities, depend upon the relative composition of the solvents, in this case, water and an organic phase. Coupled to that, there's a multi-component self-assembly process that's going on. And one of the first things one might ask is what actually goes into an mRNA. There are a relatively small number of components, all of which play an important role. Broadly speaking, what you have is an organic phase being mixed with an aqueous phase. In an mRNA vaccine, the aqueous phase contains the mRNA, which is negatively charged, and the organic phase contains the lipids that are meant to encapsulate it. And each of these plays a role. The ionizable lipids, for example, are important to neutralize the negative charge in the mRNA. Pegylated lipids are important to make these biocompatible and water soluble. Given sufficient residence and mixing time, these components mix and self-assemble to spontaneously form these aggregates. And as Matt mentioned at the beginning of the talk, the size of these aggregates oftentimes is an important factor in their pharmaceutical efficacy. So how does one go about modeling lipid nanoparticle self-assembly? I just showed you a complicated multi-component system. What we're going to do as a first step is simplify it into a one-component self-assembly model. So one-component self-assembly, I'm showing on the bottom here, where you have a set of monomeric lipids that form eventually a closed liposome or vesicle that serves as the lipid nanoparticle. And this pathway follows a sequence of steps, beginning with a fast step, nucleation, upon rapid mixing between water and organic species such as ethanol. The first step is that these lipids will spontaneously self-assemble into nuclei that are relatively monodisperse. And these nucleated aggregates subsequently grow by finding each other in the fluid space and fusing, forming larger and larger aggregates. And it's this step that is the slow step that limits the rate at which self-assembly occurs. Once these aggregates become large enough, eventually the energy required to bend a large disc-like aggregate into a closed vesicle becomes slight enough to overcome the line energy penalty for it being non-closed. And so that process is actually relatively rapid, and it results in sometimes a very broad size distribution. So each of these processes can be modeled by terms on the right-hand side of a population balance equation. What we're doing is conserving the size distribution as the aggregation distribution function, which is a function of the number of lipids per aggregate. And so the nucleation, aggregation, birth and death terms, and closure rate are captured as the source terms on this equation. Here are some of the parameters that are important in both the solvent transport and the self-assembly kinetics. The top set of parameters are familiar to anyone who has done microfluidics. The bottom set of parameters are important for the kinetics, and typically these either have to be inferred from experiment, guided by physical intuition, or turned to the literature to find some good estimates for these values. And these can vary widely depending on the particular lipid type and also the particular solvent system that you're working with. It's also useful to characterize some of the important dimensionless groups in this system. Three of them are the Reynolds number, the Peclet number, and the Dom-Kohler number. And these characterize the relative importance of inertial stresses, diffusion, and also the rate of aggregation, respectively. For typical parameters used in microfluidics, typically you're working in a modestly inertial regime and sometimes a strongly inertial regime, as Matt will discuss. Oftentimes, convection is much stronger than diffusion. And so this is coming back to the slowness of diffusion in microfluidics that I described earlier. And also, typically the convection is stronger than the kinetic rate of aggregation. So again, this is a slow step aggregation, and there's a tug of war in LNP self-assembly between the rate of mixing and the rate of the aggregation that I'll discuss in one of the case studies later in the talk.
Great, so now we'll start on case study 1. And in this case study, we'll discuss an effective type of mixing for microfluidic devices. So microfluidic devices are used to generate LNPs for small batch or personalized medicine applications. This is when you don't want a lot of a vaccine, maybe for testing, and often a lot of the components may be expensive, so smaller volumes are advantageous. The challenge, though, is that with microfluidic devices, and Joe mentioned this already, in order to achieve efficient, rapid mixing, you can't take advantage of the turbulent mixing, which is very efficient. We're stuck with the laminar flow. So one solution is to use chaotic mixing via geometric features such as the serpentine channel shown here, or active methods such as asymmetric wall motion. And all the simulations, I should say, in this case study, and in both case studies actually, were performed in COMSOL. In this section, we're using the laminar flow and the species transport interfaces. So the microfluidic serpentine mixer achieves efficient chaotic mixing through the eddies generated by the serpentine channels and the higher flow rates. The channel height in this device is 100 microns, and that typifies microfluidic devices. An inorganic, in this case ethanol, carrying the lipids, enters from one inlet, and water that carries the mRNA molecules enters from the other inlet. The flows merge, and then they're mixed by eddies, and this is visualized by streamlines colored by the ethanol concentration. The sequence of channel cross sections shows how the mixing progressively occurs along the channel. And I want to remind again that this is still laminar flow. It's just that inertia is important. Reynolds numbers are typically 220 to 100. But this mixer would not work well, at least in the context of mRNA vaccine production, at lower flow rates and lower Reynolds numbers. You need those eddies to form, and that's not going to happen in this case at the lower flow rates. Another type of microfluidic mixer is the staggered herringbone mixer, and this has a long history and it's been well published. So it's one of those mixers that when people see it, they remember it. And sets of herringbones, herringbone-shaped grooves, alternating in orientation, populate the bottom of the channel. And this causes a type of rifling of the streamlines. And because these sets of herringbone grooves, they alternate in orientation, the rifling also alternates. And so what this effectively does, it does what's called a Baker's transform on the contents of the channel. And you can think of a Baker's transform, just comes from baking. When you take a piece of dough, roll it out, fold, roll it out again, fold, roll out again. And so what this does, it takes these initially large chunks of two different solutions, and then it basically layers them progressively, more and more layers, and they fit within the same channel height, so they're closer together, and then diffusion can take over at the smaller, once they get very close and rapidly mix. So it turns out this particular type of mixer, this herringbone mixer, it's efficient across a wide range of flow rates and Reynolds numbers. So this works also at low flow rates. And in fact, Reynolds number, you could go down to Reynolds number 1. The trade-off, of course, it's a little more complicated. So there's, when you're manufacturing devices, sometimes that will matter. Also, in order to get more complicated things, you have to be very precise. So these are always trade-offs that you can consider in a design iteration. And I've just shown in this slide, just zoomed in on some of the streamlines in the lower right, colored by concentration, but you can kind of see how they move around, and that's caused by those alternating sets of herringbone patterns. So another type of microfluidic chaotic mixer uses Dean flow and splitting recombining of the flow to achieve mixing. Dean flow is a type of circulation that develops as fluid moves around a curved channel, and it's active at higher flow rates and Reynolds numbers. So once again, this particular device only works on it's still laminar flow, but the Reynolds numbers are higher, so and the flow rates are higher. As you can see, and actually you can see those recirculating patterns on the second cross section, and you can also notice that as you move along the ring after ring, the mixing gets a little bit better. We might actually want 1 or 2 more rings if you wanted to complete the mixing to a higher degree. So we've shown that effective solvent mixing can be achieved in microfluidic devices. Simulations can help tune the design parameters to optimize performance prior to fabrication and testing, and subject to a set of constraints often that have nothing to do with the fluid flow or mass transport. And we'll now show how the LNP self-assembly can be coupled to the flow and the ethanol and water concentrations.
So now we're going to show how LNP self-assembly can be incorporated into and coupled to the mixing processes that Matt talked about. So here what I'm showing is a two-dimensional model of the serpentine channel that Matt discussed in the first case study. And here, an important difference is that the flow rates here are very low. So this does not take advantage of the inertial mixing that this device requires in order to have a homogeneous outlet solvent. So as you can see, at these low flow rates, diffusion is essentially the dominant mixing mechanism. And so as a result, you have a diffuse interface at the outlet. The system is mixing limited rather than kinetically limited. The question we want to ask is, how do the flow process parameters, namely the flow rate and the ratio of the two inlet flow rates, impact size distributions of LNPs? And to model that, we again turn to the population balance equation that I introduced some slides ago. So to solve the equation in full is generally a hard problem. The first thing we can do as a first problem is make the model a bit simpler. And so we can turn to the literature for this. There's actually a very extensive literature in applying the population balance equation to problems of polymerization, coagulation and breakup, agglomeration of particles and particle manufacturing technologies, aerosol size distributions, and bubble size distributions. So one of the first papers proposing a simple model of aggregation was written in the 60s by Hilbert and Katz. The basic ingredients of this model include flow, i.e. convection of the particle size distribution, balanced by birth and death due to coagulation or aggregation. And so remember that aggregation is the rate limiting step in lipid nanoparticle self-assembly. So this is actually an appropriate first model to use for this problem. We can make the problem even simpler by assuming that the aggregation kernel is a constant that is independent of the size of the aggregates. And then this problem is actually quite tractable. In fact, we can convert this infinite dimensional distribution function space into a discrete set of nonlinear equations of finite size by simply averaging this equation, multiplying by different powers of the number of lipids per aggregate and averaging. And so what we get are a coupled set of nonlinear equations for the number density, the mean aggregation number, and the population variance. This is the so-called method of moments. And an advantage of this method is that it's computationally much more efficient than solving this integro differential equation on the top here. The disadvantage, of course, is now we're solving for statistical information rather than distributed information. And so there's a trade-off here. Another key assumption in our model is that we're going to assume that the aggregation kernel in our aggregation kinetics depends upon the solvent composition, and in fact is non-zero above the threshold water content, and so in that way couples to the solvent transport. And so in COMSOL, this is sort of how the interface would look. So here there are only 3 modules, a laminar flow and a transport of concentrated species module to account for the fluid flow and solvent transport. This can be solved on a relatively coarse mesh as a first solve or a first study in COMSOL. And then a second study, which takes information from the first, would track the evolution of the moments of the particle size distribution. And so all the population balance modeling can be done using the transport modules, using the reaction terms as a source that depends upon the solvent composition. So now I'm going to show some results. What I'm going to show on the next two slides are statistical metrics, namely the average diameter of the population at the outlet after it's been mixed in this serpentine mixer. Here it's plotted as a function of the flow rate, and the error bars in these plots indicate the standard deviation of the distribution. I want to point out that these error bars are quite large, and that's because we've assumed unbounded aggregation here. There's no closure mechanism by which these aggregates would eventually close and form and cease to continue to aggregate. As the model stands, there's only continued aggregation that's left unbound. I want to point out that since we're in a diffusion limited mixing regime, at low flow rates, you actually get very good but very slow mixing. And so the outlet size distribution has very large aggregates that are quite heterogeneous. As we increase the flow rate, you actually decrease the quality of mixing. As a result, you get smaller aggregates that follow a much tighter distribution. This is a more homogeneous population. And again, as you increase the flow rate further, eventually you would take advantage of the inertial mixing strategy of this device. We also see a dependence on the flow rate ratio. And so as you change the relative ratio of water flow to ethanol, you actually see a non-monotonic dependence on the average diameter, which is relatively interesting. This non-monotonic dependence is a result of the fact that the aggregation kinetics depend on solvent quality. And so here we're assuming a threshold water content, and that has to be adjusted based on empirical evidence as well. So this could be an example of to and fro with experiments to understand what is the optimum flow rate ratio and how does that feed into our kinetic model for aggregation. So to summarize, LNP self-assembly kinetics, we've shown a way to use population balance modeling techniques in COMSOL to couple to the laminar flow and transport of concentrated species to track solvent transport. These processes are coupled. Tractable way to handle the population balance equation is using the so-called moment averaging technique, which has its disadvantages in that you can only capture population statistics. We ran 2D simulations in a serpentine mixer in the diffusion limited regime and showed a dependence on the flow conditions, namely the flow rate and the flow rate ratio. Improvements to this model include other kinetic effects such as a closure mechanism that would bound the aggregation, and there's other different ways in which more sophisticated population balance equations can be implemented into COMSOL, and this would all be custom.